Welcome, everyone. My name is Dr. Khalid Siddig. Today, we will explore the essential principles of genetics, a rapidly evolving field that underpins modern medicine and helps us understand a wide range of diseases. We begin with the structure of DNA, the fundamental genetic material packed into chromosomes within our cells. Humans typically have 23 pairs of chromosomes, including 22 pairs of autosomes and 1 pair of sex chromosomes, XX for females, XY for males. Genes located on these chromosomes encode proteins or functional RNA. Variations in these genes can lead to disease. Genetic disorders fall into several broad categories. Mendelian disorders, caused by a mutation in a single gene, follow clear inheritance patterns. Polygenic and complex disorders, on the other hand, arise from multiple genes, often interacting with environmental factors. And then we have chromosomal disorders, caused by structural or numerical abnormalities in the chromosomes themselves. Let's go over the 4 classic Mendelian inheritance patterns. Autosomal dominant, where only one copy of a mutated gene is needed to cause disease. This pattern appears in every generation. Examples include Darie disease and Hailey-Hailey disease. Autosomal recessive, both gene copies must be mutated. It often appears unexpectedly in siblings when both parents are carriers. Conditions like albinism and beta thalassemia follow this pattern. X-linked recessive mostly affects males. Since males only have one X chromosome, a single mutation is enough to cause disease. Examples include hemophilia and G6PD deficiency. X-linked dominant can affect both sexes, but often appears more severe in males. All daughters of affected males are impacted, but no sons are. Rett syndrome and incontinentia pigmenti follow this pattern. However, real-life genetics isn't always straightforward. Several modifiers influence how these traits appear. Incomplete penetrance means not all individuals with a disease gene will develop the condition. Variable expression means symptoms can range from mild to severe, even among relatives with the same mutation. De novo mutations arise spontaneously, often in children without any family history. Mosaicism means only some cells carry the mutation. This can affect how a disease presents, or whether it's passed to children. We also must consider imprinting, where gene expression depends on which parent contributed the gene. This explains disorders like Prader-Willi and Angelman syndromes. Then there's mitochondrial inheritance, passed only from mothers. Mitochondrial DNA mutations affect energy-demanding tissues like the brain and muscles. Disorders like MELAS and LHON fall under this category. Another complexity is pseudodominance, when a recessive disorder appears dominant in a pedigree, often due to high carrier rates or consanguinity. Modern medicine relies heavily on genetic testing, including gene panels for specific conditions, whole exome sequencing, covering all protein-coding genes, and whole genome sequencing, which examines everything, including non-coding DNA. These tests are used for diagnosis, prognosis, and increasingly for personalized treatment, especially in cancer, where detecting specific mutations in tumors guides therapy. As a clinician, your role includes understanding these concepts, constructing detailed family histories, interpreting test results, and referring for genetic counseling when needed. Genetics isn't just about understanding diseases. It's about anticipating them, preventing them, and offering patients clarity. Thank you for your attention.